a popular belief that bacteria are not good for humans, that they cause infections that could prove fatal for the human body. However, what we fail to recognize is that we have many species of bacteria that reside in our own bodies in what is called the human microbiota.
The human microbiota is a densely populated microbial ecosystem that has evolved to reside in a symbiotic relationship within the human body. The gut, specifically the gut microbiota, contains about 30 to 1,000 different bacterial species. However, these bacteria do not cause us any harm, any bacterial infections. What systems has the human body evolved that allows these bacteria to reside in such mucosal environments without causing an infection? One such system is exerted by the intestinal epithelial layer. The epithelial cells of the large intestine secrete regulatory proteins into the mucosal layer, a layer that lies between the cells of the intestine and the bacterial cells. These regulatory proteins play a very specific role towards the bacterial cells. One of these regulatory proteins, RegIIIγ, protects the epithelial cells by capping the bacterial cells. This capping action occurs when they bind to the peptidoglycan layer of the gram-positive bacteria. An individual with diseases such as inflammatory bowel disease and Crohn's disease show two very distinct features in their intestines. First, the gut microbiota of such individuals is less diverse than the microbiota of healthy, age-matched individuals without any history of intestinal inflammation. Second, compared to healthy, age-matched individuals with inflamed bowels show underexpression of RegIIIγ within their intestinal mucosal layer. The intestinal epithelial cells also secrete other regulatory molecules which belong to the reg protein family. These molecules include reg1-alpha, reg1-beta, reg3-alpha, and reg3-beta. The expression of reg1-alpha and reg1-beta is induced by IL-6 and IL-22. IL-6 and 22 are inflammatory markers whose expressions are upregulated during an inflammatory response, such as during the inflammation of the intestinal epithelial cells. A specific promoter motif sequence of reg1-alpha and reg1-beta genes, called PAN1, is the binding and activation site of IL-6 and IL-22. This PAN1 sequence is not part of the RegIIIγ gene.
The first methodology involves the use of the yeast-2 hybrid assay to test genetically engineered RegIIIγ's interaction with IL cytokines 6 and 22. A positive control condition will be done by demonstrating the well-documented interactions between the native PAN1 sequences of reg1-alpha and beta with the IL-6 and 22 cytokines. A bait transcribing plasmid will be created by inserting reg1-alpha gene from mus musculus next to gal4 binding domain gene of S. cerevisiae yeast. A second prey transcribing plasmid will contain IL-6 gene of mouse inserted next to yeast gal4 activation domain gene. These plasmids will include a downstream His3 selection gene, a native yeast gene required for histidine synthesis. Plasmids will be inserted into yeast cells which are His3 oxytrophic. We should expect that reg genes with native PAN1 will result in interaction with IL-6 and 22 and thus transcribe LAC-Z and His3 in order for a blue colony to grow. In the experimental condition, the above process will be repeated, however the bait plasmid will instead be composed of genetically engineered RegIIIγ gene next to the gal4 binding domain of yeast to test whether the PAN1 inserted RegIIIγ protein can interact with IL-6 activation domain protein.
The next experiment will be done with knockout germ-free mice. Note that all mice in these groups will have GFP-tagged Reg3 protein and RegIIIγ that has been genetically modified with a PAN1 promoter region. There are two negative control groups, a germ-free mice group and a germ-free Crohn's disease mice group. These both have been induced with gram-positive bacteria in the gut with no knockout. What's positive control group will have germ-free mice and germ-free Crohn's disease mice. They both will have their IL-22 and IL-6 inflammatory markers knocked out, and they both will also be induced with gram-positive bacteria in the gut. The purpose of this experiment is to test if the genetically modified RegIIIγ under different conditions interacts with IL-22 and IL-6 inflammatory markers. This can be seen and shown through fluorescence tagging and imaging in vivo. Methodology involves using chromatin immunoprecipitation, or CHIP. The goal of using this procedure is to determine segments of the DNA that certain proteins and molecules associate with. Therefore, does IL-6 and 22 interact with the promoter motive region PAN1 on the wild-type and modified reg genes to upregulate the amount of secreted reg proteins? Two different cell lines, one with wild-type reg3 genes and one with modified version of the reg3 gamma genes, will be exposed to gram-positive bacteria. The cellular DNA will be cross-linked using either formaldehyde or UV light, giving us a control group and an experimental group. Next, a nuclease digestion process will break down the DNA to smaller segments, which will be immunoprecipitated via sedimentation and antibodies against IL-6 and 22. Finally, the DNA segments collected will be purified and sequenced to represent regions of the genome that IL-6 and 22 associate with in vivo.